So good morning, everyone. Again, my name is Remy Babich, and I'm a PhD student at Maine in the Molecular Biology and Biomedical Sciences Department. And I work with co-advisors, Drs. Nishad, Jayasundara, and Linda Silka. And today I'm going to be talking to you about some exciting new work we have been doing that couples data from the zebrafish model organism with water chemistry to better inform us of our drinking water quality, especially in the context of mixtures. So as Jane was saying, using those duplicate samples. Okay. So in this project, we are utilizing basic principles from a type of approach known as effect-directed analysis. Briefly, if we were to try to get a comprehensive analysis of the toxicity of all the chemicals in a water sample, it would be a pretty daunting task. So what we are looking to do is to reduce the complexity of what we are finding in our water samples and also limit the chance of overlooking chemicals that may pose a risk. So effect-directed analysis combines the chemical analysis of samples with biotesting, which is testing the impact of a sample, chemical, or chemical mixture on biological activity of either cells or whole organisms. And this gives us the opportunity to identify bioactive chemicals in complex mixtures. And understanding these potential driving factors is a really important step to be considered before taking management or regulatory measures. So to do this, we use the zebrafish model, which enables us to complete high throughput assessment of water samples. There are a number of attributes that make the zebrafish an excellent toxicology model, but for the purpose of this research, it's just really important to note that they are becoming a more prominent model in regards to behavioral studies and changes in behavior as a toxicological endpoint. And that's what we ended up using in this study as our bioassay. And I just have a picture there on the right of the zebrafish moving throughout early development. A goal of this project in particular was to develop an assay that would be both fast and inexpensive in determining water quality. And again, to do this, we decided to conduct a behavioral analysis after exposure to a given sample. So behavior stems from neuron development and signaling. We are able to measure changes in the neuromotor response in zebrafish larvae using the Danio vision after an exposure and determine if a chemical or chemical mixture is toxic during early development. And I have a picture of the Danio vision system that we use right here on the left. The particular focus on behavior stems from previous studies in which metals have been found to impact fish behavior. Specifically, research has drawn connections between effects of metals on development and behavioral outcomes in the zebrafish model. And as you have been hearing throughout these panels and from other speakers, we also see similar results when we look at epidemiological studies such as arsenic effects on IQ scores in children. This is a highly talked about topic both in Maine and New Hampshire. And all of these different things contribute to behavior being a really useful tool in measuring metal mixture toxicity. So now that we have some general background on both using the zebrafish as a model, as well as why we're using behavior, I want to talk in specific about the data collected and analyzed from this study, which is now in review of scientific reports. And again, I work directly here with Jane Disney and Anna Farrell, and on this All About Arsenic project. And as you may have heard from previous speakers, if you were listening in, students from Maine and New Hampshire acted as citizen scientists and collected water samples from their homes. And in some cases, 2 samples were collected. So one was sent to Dartmouth for the water chemistry report of a panel of 14 metal constituents found in the water supply, and the other was sent to me to use for biotesting. And in this case, I exposed zebrafish embryos in 50% diluted water samples in egg water from 24 hours of age until 5 days of age, at which point they're considered to be at their larval stage. I then measured this 4-day exposure impact on behavior using the Danio vision and specifically a light-dark behavioral test. The data that I extrapolated was in the form of total distance traveled over a period of 25 minutes. and changes in behavior after exposure to a water sample were compared to normal zebrafish behavior after they have been reared in the same conditions, only in egg water. So zebrafish are freshwater fish. Egg water is simply water that has undergone reverse osmosis. So we remove all the nasties, and then it is supplemented with just a little bit of salt. Therefore, embryos raised in egg water acted as our controls for this study. This allowed me to identify sample exposures that either produced no significant change in total distance traveled compared to our egg water controls. And I'll consider these samples as control-like. Samples that induced hypoactivity or a decrease in total distance traveled, or samples that induced hyperactivity or an increase in total distance traveled. And just as a side note, a lot of that work was done in the lab was with the help of an undergrad, Abigail Muscat. She was really awesome and contributed a lot to the study. So when we started looking at this data, we first wanted to look at levels of individual contaminants and how they correlated to behavior. So on the left, I am showing a heat map. The samples are broken up into sections based upon their impact on behavior. So if they were associated with no significant increase or decrease in total distance traveled, so control-like, hypoactive, or hyperactive. So for the study, we analyzed exposure to 92 samples total. 31 resulted in hypoactivity, 29 resulted in hyperactivity, and 32 samples showed control-like behavior when compared to our egg water controls. Each column within a section represents a sample. The levels of arsenic, cadmium, lead, and uranium within each sample, which are our 4 contaminants we focused on a lot in this study, just due to their known developmental toxicity, are shown as % of their maximum allowable limits, with blue colors being a lower percentage and red being at 100% of a maximum allowable limit or greater. So for example, the regulatory limit for arsenic in drinking water is 10 parts per billion in Maine, and that's what I used for this heat map. So a box that appears red in the row labeled as arsenic, or the first row of each section, represents a sample that has a concentration of arsenic of 10 parts per billion or greater. And what I really want to highlight from this is that the majority of our samples trends towards blue or cooler colors, indicating that most concentrations of these metals are within safe regulatory drinking water standards. And thinking back on some of the other plots that the other speakers were showing, we also were oftentimes seeing that trend. Now, the scatter plot to the right plots concentration of arsenic in a sample on the x-axis and the % of total distance traveled relative to controls on the y-axis. Negative values on the y-axis below -1 would indicate a decrease in behavior, and positive values on the y-axis above 1 would indicate an increase in behavior. And as you can see here, there are no apparent trends in higher concentrations being associated with a specific change in behavior. And this was pretty much seen across metals. And this really further confirmed for us that likely the changes in behavior are being driven by a mixture of chemicals rather than an individual. So to derive metal mixture effects on behavior, I combined our behavioral data with the water chemistry data and performed a principal component analysis to identify clusters of samples based upon an input of metals and their concentrations found in a given sample. So I'm going to give you a very brief overview of what I did, but this part gets a little bit heavy. So if anyone has any additional questions, I'd be happy to discuss with you further, either in the panel discussion or via chat or even in email. So in total, working from 12 metals as an input through every possible combination down to 3 metals as an input into the PCA. Again, this is from the original 14 metal panel water report. I analyzed a total of 4,016 metal mixture combinations, along with their concentrations and their relationship to behavior. This analysis produced 4,016 cluster plots Each point within a cluster plot represents a drinking water sample. Its position on the plot is based upon the metal combination input that was inserted into the PCA and the corresponding concentrations of those metals found in that sample. I could then overlay behavioral data, and from this we were able to identify if specific mixture combinations resulted in differential clustering of samples associated with behavior. I then used a quantification method modified from a study by Goodpaster and Kennedy to apply statistics to the PCA generated cluster analysis. From here, I was able to identify a subset of 2,077 metal mixtures that resulted in significant clustering between control-like and hypoactive behavior, and 193 metal mixtures that resulted in significant clustering between control-like and hyperactive behavior. And these subsets were the mixture combinations that were then used really for the rest of our analyses and what I'm going to be talking about for the next few minutes. So from this, I was able to explore which metals showed up most frequently in those mixtures that resulted in distinct clustering. Here, the blue represents percent frequency of a given metal in a mixture that was associated with hypoactivity, and red represents percent frequency of a metal mixture, or of a metal within a mixture associated with hyperactivity. So blue is a decrease in activity and red is an increase. I also looked at bi and tripartite metal mixtures, which are the graphs on the right, and their frequency within larger mixtures that were responsible for significant changes in activity levels. I did this just to try to identify if there were any small key mixture combinations we should be paying closer attention to. So some interesting findings from analyzing all of these metal mixtures and their relationships to behavioral changes include cadmium being within the top 4 metals present in mixtures resulting in both hyper and hypoactive behavior. Lead was found in 67% of the metal mixtures that were associated with hypoactivity and was present in 10 out of the 15 small mixtures found associated with hypoactivity. Uranium was in 45% of metal mixtures associated with hyperactivity. Also, uranium and antimony were the top bipartite combination found in metal mixtures associated with both hyper and hypoactivity. And arsenic was present in 50 and 38% of metal mixtures responsible for hypoactivity and hyperactivity.
About 2 minutes left, Remy.
Okay, thank you. So some takeaways from this work: zebrafish can be used as a biological tool to better understand our water quality. Citizen science allowed for the collection of a large number of randomized samples and created conversations around the importance of water quality among local students, teachers, and families. And, you know, I think we're really just starting to skim the surface on this. And even the work that was, you know, just put into this project so far by our lab, you know, we can do so much more with that data. So one of the next things that I'm really looking to do is get this data into an appropriate format for TUVA and let the students really manipulate it and make their own discoveries. I think that can be really great. And also, low-level metal mixture effects should be taken into consideration when determining water quality standards, especially here in the case of arsenic, cadmium, lead, and uranium. And also, I think this really couples hand in hand with what Sarah Hall was talking about with changes in seasonality and, you know, how these different metal concentrations are going to be potentially going up and down, and their biological impacts when that happens. So with that, I would just like to thank everyone, our funding sources, thank MDI, Jane, Anna for having me be a part of this project. And also a huge shout out to all the teachers and students involved. I wouldn't have been able to do this work without your participation, and I think it's really great. So thank you.